Good morning, everyone. I am Virgilio Ebajo Jr., the equipment specialist for the NMR laboratory located at the DLSU Laguna campus. And my task this morning is to present to you some researches utilizing nuclear magnetic resonance spectroscopy to address the UN Sustainable Development Goals. In 2015, the United Nations General Assembly adopted a new Sustainable Development Agenda which includes 17 Sustainable Development Goals to end poverty, protect the planet, and ensure prosperity for all. If achieved, these 17 goals will fundamentally change the world for the better. Chemistry offers a broad spectrum of products and services essential to our daily lives that are safe, sustainable, and environmentally sound. Chemistry also enables more efficient use of our natural resources, increases energy efficiency, allows for reduced greenhouse gas emission, development of new medicinal drugs to fight diseases, and is at the forefront of development of sustainable materials. In short, chemistry is going to have to play an essential role if we are to meet the UN Sustainable Development Goals. Chemistry is obsessed with identifying structures at the molecular level. We always want to figure out the type of bonds present in a molecule, how atoms are connected, or how molecules are arranged in solid state. This obsession is due to the fact that difference in connection of atoms, difference in bonds, or difference in crystal arrangement have a profound effect on the properties of chemical compounds. For example, hydrogen gas, composed of two atoms of hydrogen covalently bonded to one another, is a highly flammable gas. On the other hand, O2, oxygen gas, composed of two atoms of oxygen, is an oxidizing agent. Both molecules are ingredients in making fire. But if you take one atom of oxygen and put it in between hydrogen atom in H2, you will end up with water, a molecule that can extinguish fire and is also necessary for life on Earth. At the heart of every chemistry research institute is the very powerful instrument that can help determine molecular structures, the nuclear magnetic resonance spectrometer, or what we also refer to as the NMR. Since NMR is the topic of my talk, it is proper to revisit the basic principles of NMR spectroscopy. The NMR spectrometer is a system consisting of a superconducting magnet, the NMR probe, the preamplifier and spectrometer, and the desktop computer. The computer controls the whole system by sending commands to the probe and spectrometer. The spectrometer carries out the calculations and sends the appropriate energy to be used by the probe and the preamplifier. The probe serves as both the detector and source of energy in the form of weak radio frequency pulse. The NMR spectrometer lies on the NMR theory, which involves the interaction of a nucleus with spin, a strong external magnetic field, and a radio frequency pulse. A spinning nucleus possesses a magnetic field and it acts as if it is a tiny bar magnet. And when introduced to a strong magnetic field, these tiny bar magnets align themselves either parallel or opposite to the direction of the external magnetic field. Upon the application of a specific RF pulse, the spin absorbs energy and transitions to an excited state by flipping to a certain angle. Usually, this is a 90-degree angle. By detecting the absorption signal, one can obtain a free induction decay, a data in the time domain, which can then be transformed to an NMR spectrum after the application of Fourier transform. The NMR spectrum do not give a direct picture of the structure of molecule. It only contains signals that we call peaks. The location and the fine structure of the signals dependent on its chemical environment, while the area under each signal is dependent on the number of nuclei experiencing similar chemical environment. By careful interpretation of the location, area, and fine structure of the signals, chemists can determine the structure of molecules. NMR is considered as the modern age lab thermometer, which means that every chemistry research institute should have one, or at least have access to it. Fortunately, De La Salle University currently has an NMR spectrometer housed in the NMR laboratory of the Central Instrumentation Facility. located in the SU Laguna campus. In the Nuclear Magnetic Resonance Laboratory is the 600 MHz GLECCR Nuclear Magnetic Resonance Spectrometer. With the 14 Tesla superconducting magnet, this is the most powerful NMR spectrometer in the Philippines. It is equipped with a two-channel amplifier system, which allows it to execute multiple complex multiple pulse experiments. The 600 MHz gel NMR spectrometer is equipped with two probes, the liquid or the solution probe and the solid probe. This allows us to conduct NMR experiments in both liquid and solid state. At present, we are the only laboratory in the Philippines capable of performing solid-state NMR analysis. We offer the following NMR analysis in our laboratory. Routine 1D NMR analysis, such as proton carbon. Complete 2D, two-dimensional NMR analysis for structure elucidation. Multinuclear NMR analysis focusing on NMR active nuclei, aside from carbon and hydrogen. So these are the chlorine-19, phosphorus 31, silicon 29, up to nitrogen 15 frequency range. Multi-pulse experiments, variable temperature experiments for kinetic or stability studies. The temperature can range from negative 100 degrees Celsius to 150 degrees Celsius. Solid-state NMR analysis of powder samples, and also copolymer analysis. If you are not familiar on how to read NMR results, or you would like to improve your knowledge on NMR analysis, we strongly recommend that you avail of our workshop or training services for basic NMR spectroscopy. If you are in the field of natural products and you want to learn how to interpret NMR spectra in order to determine the structure of your isolates, you can take our basic and advanced NMR spectroscopy for structure elucidation training service. Now that I have already introduced the NMR instrument and its working principle, the next question that you might have in mind is how can NMR help attain the Sustainable Development Goals? On the list of 17 SDGs, the NMR shines on the categories of good health and well-being and affordable and clean energy. The development of novel drugs to fight new variants of diseases is one way of addressing good health and well-being. Nature is the best organic chemist. It is a gold mine of thousands of diverse organic compounds, some of which are capable of curing health problems that the society is facing. A field of chemistry called natural products chemistry is devoted in finding novel organic compounds produced by living organisms in order to find potential medicinal drug candidates that could help the humanity. Natural products researchers aimed at finding novel drug candidates from plant secondary metabolites often follow this bioassay guided extraction protocol. This process starts with the identification of plant material. Most of the time, the plant candidate is one that is known to be a folk medicine or a plant that have zero studies. After the plant is identified, the plant parts will be subjected to solvent extraction using solvents of varying polarity. These extracts are called crude extracts because it's still a mixture containing dozens of compounds. Each crude extract will be subjected to a certain bioassay. For example, cytotoxicity assay if an anti-cancer compound is desired. The crude extract that gives the best result in the bioassay will then be subjected to fractionation using chromatographic techniques. In our research, we often use the traditional gravity column chromatography. Different fractions will be collected. Each fraction now contains lesser number of secondary metabolites compared to the crude extracts. The fraction will be subjected to bioassay, and the fraction with the best bioassay result will be further fractionated to give fractions with lesser amount of secondary metabolites. This fraction will again be tested using bioassay, and the fraction with the best activity will be further fractionated via chromatography. The process of fractionation and bioassay will be repeated until the biologically active fraction is pure and contains only a single secondary metabolite. This fraction should have a single spot on its thin layer chromatography profile. The compound present on the pure fraction will then be analyzed using NMR. And after a careful interpretation of the NMR spectra, the structure of the compound causing the biological activity can be identified. Once the structure is determined, several tests and trials will then follow for the compound to be commercially available as a drug for treatment of the target disease. As an example of success story for natural products chemistry research programs is the isolation and structure determination of vincristine. Vincristine was isolated from Madagascar periwinkle Catharanthus roseus. It is a plant used as a folk remedy for centuries. It is now commercially sold under the brand name Oncovin and is used as chemotherapy medication for different types of cancer. Another success story in natural products research is the isolation and structure isolation of paclitaxel, which is derived from Pacific U3. Paclitaxel, now commercially sold as Taxol, is used as treatment for different types of cancer. Extraction of paclitaxel from the Pacific U3 for commercialization is not sustainable because 1,200 kilograms of tree bark produces only 10 grams of pure paclitaxel. So paclitaxel sold in the market are not extracted from plants but are produced in the lab by synthesizing from smaller and cheaper starting materials. The industry scale synthesis of paclitaxel and vincristine will not be possible without the identification of its molecular structure. In this aspect, nuclear magnetic resonance spectrometer played a very important role. Philippines is listed by the World Conservation Monitoring Center of the United Nations Environment Program as one of the 17 mega-diverse countries in the world. A large percentage of natural flora and fauna in the world can be found in the Philippines. There's also thousands of plants that are endemic or can only be found in Philippines. So who knows, maybe the next anti-cancer compound is in a plant that is endemic to our country, waiting to be discovered by our natural products researchers. In order to determine the structure of this compound, you need to make use of nuclear magnetic resonance spectrometer. Access to good quality drugs in the market will ensure good health and well-being. Different analytical techniques can help assess the quality of drugs. In drugs existing in solid states, such as tablets and powders, a common problem encountered is the formation of undesired crystalline form. This can only be identified by solid-state analytical techniques such as solid-state nuclear magnetic resonance spectroscopy. Most pharmaceutical products are formulated with excipients and drugs as solids in the crystalline states, and developing molecular crystals in various dosage forms is preferred for stability and manufacturability reasons. Generally, drug molecules are more chemically stable in the solid state compared with solution, where degradation occurs more easily and readily. This, however, does not suggest that pharmaceutical solids do not present their own challenges. In the solid state, molecules can adopt more than one packing arrangement and or conformation in the crystal lattice, which give rise to the phenomena of polymorphism. Polymorphism, as an example, shown below is the polymorphism of ranitidine. So ranitidine can exist in two different polymorphic forms called form 1 and form 2. They are product of different recrystallization process. Form 1 and form 2 have the same molecular structure, but their crystal orientation are different. Form 2 is characterized with its larger and more ordered crystalline state, which gives it a better flow property during manufacturing. The way a molecule packs in a solid influences different physical and chemical properties, such as solubility and dissolution rate. Now, solubility and dissolution rate are very important properties in orally administered drug products. And polymorphs of a drug with low solubility and dissolution rate will often result to a poor performing drug. A well-known case of polymorphism leading to the formation of inactive drugs was reported in Kenya, Africa in 1999. Helminthiasis is caused by a group of parasite commonly referred to as worm, and the most common treatment for helminthiasis is the deworming drug mebendazole, and the World Health Organization recommends the annual or biannual deworming using this drug in order to prevent helminthiasis. Now, in African countries, helminthiasis is prevalent, and to address this issue, a deworming program was conducted in Kenya using mebendazole as recommended by WHO. However, um, mebendazole exists in three different polymorphic forms, Form A, Form B, and Form C. And only Form A is active against helminthiasis. Form E, B, and Form C are inactive. There were reports in Kenya of mebendazole being effective, and researchers in Kenya found out that most of the mebendazole raw materials imported by Kenya exist in the inactive forms B and C. This leads to a call for better quality control of mebendazole production in Kenya. This event shows that polymorphism in medicinal drug is a serious problem, and there should be a fast and effective way of determining drug polymorphic forms. Now, solid state nuclear magnetic resonance spectroscopy is a very effective tool for determination of drug polymorphic forms. The power of solid state NMR in polymorph analysis can be seen in the analysis of the indomethacin polymorphs. Indomethacin is a commercially available antipyretic and anti-inflammatory drug, and there are two well-known polymorphs for indomethacin, the alpha and the gamma forms, which varies on their stability. The two polymorphic forms cannot be distinguished from one another using solution state analysis, such as the solution state NMR. As you can see here, both the alpha and the gamma form have very similar carbon-13 NMR spectra. You cannot tell one from the other by just looking at their carbon-13 NMR spectra. However, when the samples are analyzed using solid-state NMR analysis, the two polymorphic forms showed different carbon-13 NMR spectra as shown here. The alpha form have more peaks that are splitted, and while the gamma form have peaks that are wide and are singlet. This shows that the solid-state NMR is an effective tool for identifying drug polymorphs, which can be used to ensure that drug products being produced are of desired polymorphic form. Now, two of UN SDGs, which are affordable and clean energy, and climate action aims at avoiding dangerous climate change by limiting global warming to well below 2 degrees Celsius. Energy storage in secondary or rechargeable batteries is a versatile and lower emission option to achieve the aforementioned goals. Battery is one of the most important inventions of mankind. It is one of the products of science that we use every day. For example, our smartphones and laptops relies on energy stored in batteries. At the forefront of battery research are lithium-ion batteries or LIB. And in the past decades, the research on secondary LIBs has been driven by the increasing demands of portable electronic devices. LIB typically is composed of carbonaceous um, anode, a transition metal oxide cathode, and they are connected with a metal plate or wire as current collectors. It also contains electrolyte solution containing lithium salt and an organic solvent. Organic solvents for LIB are typically ethylene carbonate, dimethyl carbonate, and diethyl carbonate in either pure form or as mixtures. I will not go into detail of battery chemistry, but it's important to note that during charge-discharge cycle, the lithium ion flows from one electrode to the other, resulting to a generation of current. Since the charge-discharge cycle of a battery is an electrochemical process, it is important that the electrolyte components remain stable um, during long runs. Now, since the electrolyte solvents are organic compounds, their proton NMR can be used to monitor their stability. The proton NMR spectrum of the electrolyte comprising of 1 molar of lithium hexafluorophosphate in a mixture of ECDMCDEC 1:1:1 by weight showed well-resolved and separated signals of the organic carbonates as shown here. You have here the signal for DEC, the signal for DMC, and the proton NMR signal for EC. Now, after 1 week of storage of the solution, the electrolyte solution containing the mixture of organic solvents and the lithium hexachlorophosphate, uh, the proton NMR shows that there are new signals that are not coming from the um, pristine solvents. So this means that during storage at 85 degrees for 1 week, the organic solvents decompose, resulting to the generation of new signals in the NMR spectrum. Now, the inorganic components of the electrolyte solution containing chlorine and phosphorus can also be characterized using chlorine 19 NMR and phosphorus 31 NMR. So you have here phosphorus 31 on top and the chlorine 19 NMR spectra at the bottom. It is shown here that just like the organic components, the lithium hexafluorophosphate also decomposes after long storage, leading to the formation of different compounds as observed by the generation of the new signals here in the phosphorus 31 NMR, ranging from 0 to negative 25 ppm. And in the fluorine 19 NMR, there are new signals from ranging 5.2 to around 5.75 ppm. And these are signals coming from degradation product, and they are not due to the lithium hexachlorophosphate present in the electrolyte. Now, degradation of electrolyte may lead to undesired events such as short circuit, which in the presence of the usually flammable electrolyte could cause thermal runaway, for example, fire and explosion. Of course, everyone's aware of the Samsung Note 7 battery explosion. And the battery in Samsung Note 7 is a lithium-ion battery. So in order to prevent failures and improve lithium-ion batteries, it is strongly recommended to include the NMR analysis in the research and development process or R&D stage of LID generation. Now, other components of the LID, such as the carbonaceous anode and the transition metal oxide cathode, can also be analyzed using solid-state NMR to gain insight on their stability. So, in theory, you can analyze the whole components of battery using NMR. Now, difference in synthesis procedure in different laboratories may also lead to formation of different crystalline forms of the anode or the cathode material, leading to varying quality of LID, and in some laboratories, it may lead to low quality LID. It is therefore recommended to have the LID analyzed during the R&D development in order to prevent failures in the future. In summary, I have presented researches on drug discovery, drug quality, and lithium-ion batteries, utilizing the nuclear magnetic resonance spectroscopy. These are just some of the examples of how NMR can be used in attaining sustainable development goals. Now that we have access to this very powerful instrument capable of determining molecular structures, we can now level up our research and start looking at what is happening at the molecular level. We are working on researches which wherein you think NMR can help you address some of your problems. Feel free to email us at nmrlab@elsu.edu.ph. We would be very happy to collaborate with you and help you in solving your problems. Thank you for your time and attention, and we are looking forward to working with you.